Okay, so last class we finished off with chapter 4 and 5. Today we are going to be starting off with chapter number 6, that is your plant nutrition. So we are talking about plant nutrition. Okay. So firstly, if I talk about, plants making their own food, plants are autotrophs and they make their own food. They make organic nutrients from inorganic substances. Basically, they use carbon dioxide and water to produce glucose and oxygen in the presence of sunlight alongside chlorophyll, and this process is called photosynthesis. Right? So they use carbon dioxide and water to produce oxygen, glucose and oxygen basically in the presence of sunlight with chlorophyll. Okay, this process is photosynthesis. Again, if I talk about the chlorophyll is the green pigment. Okay, this is what gives chloroplast the color. Okay, traps sunlight and converts light energy to chemical energy. So chlorophyll is the green pigment. It's the green pigment that traps sunlight and converts light energy into chemical energy. Alright, if I talk about the equation, how do you write it? First, let's talk about the symbolic equation. It's a reversible reaction, obviously. You have sunlight and chlorophyll. Now, these sometimes they ask you what is on the arrow, sometimes they don't. So if they don't write it. If they do, then you know what is on the arrow. This was a symbolic equation. Now, word equation: carbon dioxide plus water gives you, sorry, glucose and oxygen. Okay. Now, obviously, plants are making carbohydrates. So obviously, there will be some usage of this carbohydrate. So this glucose is a carbohydrate, right? So plants will obviously have some usage of it. Now, if I talk about here, one second. Usage of carbohydrates in plants. Okay. Firstly is releasing of your useful energy. Okay. You know that some of the glucose is used by the plant itself, various activities. Obviously, they respire as well, right? So energy is released from glucose by respiration. Just like we do. So similar to that, plants also do that. Then it's storing to use later. So obviously excess glucose by plants that is made is stored by converting it into starch in the form of starch. Remember this. Be stored in the form of glycogen. Okay. So conversion of glucose into starch is important because starch molecules are insoluble in water, so they do not get involved in any of the chemical reaction taking place in the plant cell. They do not affect the concentration of solution inside the cell. Okay, and starch molecules can be broken down very easily into glucose back. Okay, so there's no issue. Third is making sucrose for transport. Making of sucrose for transport. Okay, so sucrose is also another type of carbohydrate. Okay, plants transport glucose from the leaf to other parts in the form of sucrose via phloem. And why is it in the form of sucrose? Because again, sucrose is insoluble, and if you guys know, phloem is made up of living cells, right? Your yes. So if your glucose will be transported in the same form, okay, in the form of your glucose, it's not converted into sucrose. What will happen? Your phloem cell will utilize it. There will be no point of transporting it for storage. Okay, that's why we transport it in the form of sucrose so that your phloem cells cannot utilize sucrose because it's a large molecule. Okay, it's insoluble, it's large, so it cannot be consumed by the phloem cells. That's why we convert it in the form of sucrose and then transport it. Okay. Fourth is making of cellulose to build cell wall. Okay. So growing plants, they all need a cell wall, and cell wall is made up of like links of glucose that all link together to form a long chain, and then that's makes cellulose. Okay, it forms fibers. There are cross links that develop. It basically provides it the strength. Then they can also, these are the main things. Now they can also make nectar to attract pollinators. Nectar is also another form of carbohydrate. Okay, it's sweet. So making nectar to attract pollinators. So you know that flowers produce nectar that animals can feed on. Okay, so in the meanwhile, while they're feeding, pollen can get attached to them. It can stick them on them, and then pollination can take place through your pollinators. Then they can also make amino acids to make proteins. So plants can also use glucose to make amino acids. Okay, so these amino acids can then be used to make proteins for growth. Not all of the glucose is stored and utilized. Some of it can also be used to make other substances like amino acids because proteins are also important. Proteins make enzymes. Proteins make the structures. Okay, so proteins are also very important. So glucose can also be used to make amino acids that can further make proteins. And then there are some other substances. For example, chlorophyll. Your glucose can also be used to make chlorophyll because without chlorophyll, your leaves will turn yellow. There will be deficiency of chlorophyll. Okay, basically you need nitrate and magnesium ions for chlorophyll, but glucose also providing them energy. You need nitrate and magnesium ions. And if you don't have enough chlorophyll in the leaf, it will turn yellow. Is this clear? The usage of carbohydrate? Anyone, any questions?
Glucose is converted to starch in chlorophyll.
No, no, not chlorophyll is for trapping sunlight. Glucose is not converted to starch in the chlorophyll. It is stored in the roots in the form of starch. Chlorophyll is for trapping sunlight. Okay, sir. Chlorophyll has nothing to do
Yes,
with.
clear.
I think you're talking about starch grains, those small starch grains, right? You're talking about that.
Yes,
Okay, yeah, that is there. So see, mainly it is stored in the form of roots in large quantities. Okay, and very minute
yes, clear.
quantity. Minute quantity is those. I don't you can't even see those starch grains in your chloroplast. I'll draw the structure and then you will see. So very minute quantity is in the chloroplast. If it is in excess, rest main storage is in the roots. Okay, like potato tuber. I hope you remember when we talked about in the plant reproduction. I don't know if you were there, right? So potato tuber, that is also an example, right? They grow underground, right? So roots store your starch. Is this clear?
Clear.
Okay. Anyone else? Any questions? Everyone's clear? Ayla, Ahmed, Zoya, Abdulbasit.
Yes.
All right, great. Please note this down. Then we'll talk about the structure of leaf. Making other substances. See, you need to absorb nitrate and magnesium ions from the soil, and for that process, active transport will be required, and active transport requires energy, right?
Okay.
Right. Okay. Are you guys done?
Done.
Rest of you? Okay, then let's move on. All right, great. Let's move on then. Is the basic clear? Any questions?
No, miss.
Okay. Now let's talk about the structure of a leaf. I guess if I draw a leaf, okay. Let's take it. Please. Okay. This part is called your leaf blade or your lamina. These are your veins, obviously. This is your midrib. And this is your leaf stalk. Right now it looks quite simple. And we're like, chloroplast is present and chlorophyll, chloroplast is present, chlorophyll is inside there, photosynthesis takes place here. But if you look at it in detail and talk about the tissues in a leaf, one second, I have a diagram, just copy it. Just share it back the screen. All right, here. Okay, there. This is what the tissues look like. Okay. The top is your upper epidermis. They haven't mentioned it. You have cuticle on top of it. This layer is your cuticle. You see this layer, this one. So cuticle. Okay. This is your palisade mesophyll layer. It's also called palisade mesophyll. And the bottom one is your spongy mesophyll. These are vascular bundles, xylem and phloem. Lower epidermis, you have stomata. You have guard cells. Alright, so the top of the leaf is your upper epidermis. And they contain like yes. Alright. Okay, now look at the diagram while I'm explaining. So top of your leaf, you have your upper epidermis. Oh, what is the upper? You have the upper epidermis. Okay. Cells are tightly packed. Okay. And why is that? It's to reduce the quantity of water vapor escaping from the leaf. Okay. Now they cannot photosynthesize. Why? Because they do not contain any chloroplast. And these cells secrete a waxy cuticle that is your present on top of your upper epidermis, as you can see the diagram. Okay, so this basically forms a thin, transparent, waterproof covering. Is this clear? It forms a thin, waterproof and waterproof covering, transparent as well. So this cuticle also prevents loss of your water through the evaporation and acts as a physical barrier to prevent the entry of pathogens as well. Right? So it's thin, transparent, and waterproof. Okay, it prevents the loss of water through your evaporation and acts as a physical barrier to prevent your entry of pathogens. Then below the upper epidermis, you have a palisade mesophyll layer or palisade layer. Okay, it is made up of long, narrow cells. Can you look at them? And that contain numerous number of chloroplasts because this is the point where maximum photosynthesis takes place. They receive maximum sunlight. How? Now see, cuticle is transparent. Upper epidermis is just one layer. So the sunlight comes directly, goes through cuticle because it's transparent, and then the upper epidermis. So maximum sunlight is reaching your palisade mesophyll cells. That's why this layer contains the maximum number of chloroplasts, so that maximum amount of photosynthesis can take place at this point. All right. So beneath the palisade mesophyll cell is your spongy mesophyll layer. Okay, so your spongy mesophyll layer, and these cells in the layer are round. They're not tightly packed. Okay, and have many air spaces between them, as you can see in the diagram as well. Okay, they do contain chloroplasts, as you can see in the diagram, but they do not have as many chloroplasts as in your palisade mesophyll cells. Okay, because this layer is not made for photosynthesis. The main function is gas exchange. That's why they have spaces between them. Okay, that's why it's called your spongy mesophyll layer. Okay, spongy mesophyll cells are here. Okay, yes, there are chloroplasts because it does perform photosynthesis, but point is main function is not performing photosynthesis. Main function is your exchange of gases. All right, then you have your lower epidermis, and that is similar to that of your upper epidermis, but point is, except for the fact that there is no waxy cuticle present, because at the lower part it will not get as hot because sunlight is not, you don't get sunlight on the lower part of the leaf, right? So there are openings in the lower epidermis that are called your stomata, as you can see in the diagram, this part. Okay, and they are this stoma is surrounded by a pair of guard cells. Opening and closing of guard cells, this when guard cells open, it forms a space that is called your stomata. Guard cells close, stomata closes. So opening and closing of guard cells, how they move is basically dependent on your calcium ions. Okay, and when your water is moving inside, okay, cells swell. Okay, so guard cells change the shape, which can open and close the stomata, and stomata then allows the diffusion of carbon dioxide, water vapor, and oxygen in and out of the leaf. Are all the layers clear?
Do we need to memorize the working of guard cells?
Working of guard cells, just remember that when they change their shape, that opens and closes stomata. Remember that stoma is surrounded by your guard cell. Okay, and the fact guard cells also contain chloroplast. That's the working. What else? Nothing else. You don't learn anything else. Okay.
What is the difference between the purpose of the cuticle and the upper epidermis?
Between structure of cuticle and upper epidermis.
Yes.
See, upper epidermis secretes cuticle. Cuticle is not already there. Upper epidermis secretes it. Okay, upper epidermis is already part of your tissue. So basically, imagine if upper epidermis was not secreting the cuticle, our leaf tissues layer would have started from upper epidermis. Okay, upper epidermis is part of it. Upper epidermis is basically, it's just a layer, like you have skin, and imagine if I have skin and I put Vaseline on it. Okay, then you ask me difference in structure of Vaseline and skin. Okay, skin is something I put on my own, right? And skin is obviously to protect my, it's like it's in covering, right? But in this case, cuticle is secreted by your upper epidermis. The cuticle basically just is there for making it waterproof. Okay, not, it's basically also acting as your what, physical entry, basically path. It provides your physical barrier to your pathogen. Okay, is this clear? Upper epidermis is just there, it's like the first layer of your leaf. Alright, so first
Yes,
is your
clear.
epidermis. Okay, good. Anyone else, any questions?
No.
Rest of you, Zaya and Emma, the Bassett.
No, miss.
Okay, great. Upper epidermis is there and it secretes cuticle. Waxy cuticle, okay. This waxy cuticle basically is thin, transparent, and waterproof. Okay, and basically acts as physical barrier. Prevent entry of pathogen. Alright. Okay, no photosynthesis takes place here. Then I have second palisade mesophyll layer. Palisade mesophyll layer. Okay, it contains numerous number of chloroplasts and maximum photosynthesis takes place here. Numerous chloroplasts and maximum photosynthesis takes place here. Third, spongy mesophyll. Okay, so beneath the palisade mesophyll is the spongy mesophyll layer. Cells in the layer are round, not tightly packed, and have many air spaces between them. So you'll say this is gas exchange, main gas exchange layer. Okay, because oxygen, carbon dioxide, water vapor diffuses from here. Then I have lower epidermis. Okay, so it does not have any chloroplast and all. Okay, it has stomata, which is surrounded by guard cells. By a pair of guard cells. Okay, guard cells change shape to control the opening and closing of stomata. Which control the opening and closing. Which open and close the stomata. Okay, now if I talk about your chloroplast. This is what it looks like. Okay, so these were the starch grains you were talking about, right? Very minute quantities of starch grains are in your chloroplast. Okay. Alright. So you have ribosomes for protein synthesis free DNA. This is a stack of membrane that contains a chlorophyll. Okay. And this is outer membrane, inner membrane is double membranous organelle. You know that. Right, these are your stock particles. Okay, your lamellae and all. We are going to discuss in detail. And you're going to discuss this in A levels, inshallah, in more detail. Alright, now let's talk about your factors affecting your rate of photosynthesis. Firstly, you need to, before discussing the factors, you need to know what exactly is a limiting factor. Limiting factor is any factor that is in short supply and therefore limits the rate of photosynthesis. Is this clear? Note this down first, and then we're going to discuss your factors affecting rate of photosynthesis. Alright. Rest of you done?
Yes.
Saad, Jabasit, Ahmed.
None.
Okay, let's move on then. Now we are talking about your factors affecting photosynthesis. Okay, so there are several environmental factors that affect the rate of photosynthesis. For example, firstly, you have supply of raw materials. Okay, raw materials include carbon dioxide and water. Secondly, you have the quantity of sunlight. Basically, it is the light intensity. One second, let me reshare the screen. Here. Let me reshare the screen. Quantity of sunlight, basically because that provides energy for the rate of reaction. Then you have temperature. Because that affects the activity of enzyme. This also you can see provides energy for reaction. Affects activity of enzymes. Okay. First, let's talk about carbon dioxide concentration. So more the carbon dioxide a plant is given, faster it can photosynthesize. But once it reaches a certain level, there is no further increase in the rate of photosynthesis. Why is that? Because now there is some other factor that is affecting the rate of photosynthesis. Carbon dioxide is no longer the limiting factor. Okay. As it increases, no less carbon dioxide concentration increases. Rate of photosynthesis also increases. But after a certain level, once it reaches a certain level, but after a certain level, does not increase. As some other factors is now limiting the rate of reaction. Is now limiting the rate of photosynthesis. Let me just copy a graph, then I'll show you. 1 second. Oh, look at this one. Okay, yes. Okay. Right, so you can see rate initially increases and then after a certain point does not increase because now some other factors affecting the rate of photosynthesis. Now, in your carbon dioxide concentration, stomata will play a role. Okay. 1 second. So stomata plays an important role because carbon dioxide diffuses into the leaf through the stomata. Stomata are closed. If they are closed, photosynthesis cannot take place because plant requires one of the raw material. Okay. Actually, this should have been the first one. Anyways, I'll shift it. No, right. If you haven't written, please write stomata first. Okay. Or if it's otherwise, number it. This is number 1 and this is number 2. I'll just shift it. So carbon dioxide diffuses into the leaf. And if it is closed, photosynthesis will not proceed. As a raw material is missing. Okay. Let me just shift this. This should come at the second number. First, I wrote about raw material. Raw material, right? 1 second. Okay. This is first. Second. If you've already written it down, please change the numbers. Okay. Make that number 1 and then make this number 2. I have switched the order. Okay. Carbon has concentration. Yeah, because it's in raw material, right? So it's the same thing. Okay, now we talked about that. Now let's talk about sunlight. So we'll talk about your light intensity. These are in the same category, supply of raw materials. Okay. Okay. Now let's talk about light intensity. So light intensity increases, just like carbon had concentration. Rate of photosynthesis also increases. Light intensity increases. Rate of photosynthesis also increases. Rate of photosynthesis also increases. Alright, until it reaches the maximum rate. And after that point, and after this point, even if light intensity increases, rate of photosynthesis will not increase. As plants, as there is some other factor. What did I write here? As some other factor is now limiting the rate of photosynthesis. Is now limiting the rate of photosynthesis. Okay, let me copy a graph and show you how it looks like. 1 second. Okay, right. So this is what it looks like. You can see it increases, and now light intensity is no longer a limiting factor. Okay. So then last is your temperature test. This was third. So some of the reactions taking place in photosynthesis. Some of the reactions taking place in photosynthesis. Photosynthesis. Photosynthesis are catalyzed by enzymes. Hence, temperature affects the rate of photosynthesis. Okay. So similarly, temperature increases, rate of photosynthesis also increases. Rate of photosynthesis also increases. Okay. And again, this is also until a certain point, because photosynthesis involves enzymes and they can get denatured at high temperature. We talked about this yesterday. Okay, and we can say this like we talked about carbon dioxide, but after a certain level, also increases. This is sorry. This is up till a certain point. And why is that? Because photosynthesis has sorry has sorry involves enzymes which can get denatured at a high temperature. And we have seen this yesterday. Again, same phenomena will be followed. Kinetic energy increases. That's why rate of reaction increases. Molecules collide faster. More enzyme substrate complexes are formed. Or this is only up to a certain point, because after that your enzymes get denatured at high temperature. Alright, is this clear? Any questions, anyone?
No.
Alright, rest of you. How will we know exactly what factor is limiting after the certain point? Then this can be done practically. Then start to increase one factor by other, one by one. First to increase carbon dioxide. If rate increases, well and good. If not, some other factor. This is done practically. You cannot tell this theoretically. Okay, sir. Zoya, clear? So you figure it out by doing this practically. All right. Zoya, clear? So if this question is asked, can I add any other factor? Yes, you will say that now some other factor is the limiting factor, such as carbon dioxide concentration, temperature. Okay, there's also humidity. Okay, at times, if you talk about that will be in the rate of transpiration. But so you can write some other factors now, the limiting factor, and then you can give 2, 3 examples. They won't ask exactly, no, they will never say exactly which factor. Okay, either you can say some other factor, you can give 2, 3 examples. You cannot, I'm telling you cannot figure out which factor exactly. How will you just by looking at the graph tell which factor is not affecting? Unless this, say in the question that these two factors you are experimenting. Okay, it depends on what type of question is there. Okay, if they're saying there's experiment being carried out in which your temperature and carbon dioxide concentration are being figured out. Okay, now in this graph we are increasing light temperature, as they have given you a graph and they're increasing temperature. And now they are saying that after a certain point your graph flattens off. Now which factor could be involved, could be responsible for limiting the rate of photosynthesis? Now if they have given you the question, then you will say yes, carbon dioxide concentration. Okay, because they're telling you that there are two factors that are being involved in the investigation. If they're not, then it's open-ended. You cannot tell without performing it. Okay, this is nothing. This cannot be answered theoretically. You need to perform it. No problem. Anyone else? Any questions? Saad, Dubasit, all clear?
Yes, miss.
Okay, please note this
Yeah.
down. Okay, great. Note this down, please. Tell me when to scroll down. Not exactly. You can, figure that out with your enzymes. It's the same thing, because we're talking about the rate of enzymatic activity, right? Enzymes get denatured. So we did that yesterday. So you can refer to the same graph, because temperature's effect we talked about with respect to affecting your enzymes. That is what in your syllabus. Okay, so you can refer to the same graph. Okay, great. Okay, I need to ask you guys one thing, and I need class response on that. Now, according to your books, okay, books first cover plant nutrition, then they cover human nutrition, and then they cover transport in plants, and then transport in animals or humans. Okay. When I first taught the course, I did plant nutrition with transport in plants. So, for example, chapter 6 with chapter 8. And then I did human nutrition and then transport in animals or humans. Chapter 7 with chapter 9. So, should I follow the book sequence? Like, first, we are done with plant nutrition today. Tomorrow, should I do human nutrition or go according to what we are studying, plants, and do transport in plants? Yes. Tomorrow, should I do human
Transport in
nutrition?
plants.
Okay, transport in plants. Okay, rest of you. Because see, my point is, yes, exactly. See, point is that we are studying plants, right? So it's better once we are done with plant nutrition. Now we know how we transport the substances made from it. Okay, transport in plants, transport in plants, transport in plants. Okay, 3 votes I got for transport in plants. Okay, Ahmed, Abdul Basit. Yes. Oh wow, transport in plants. Nice. Ahmed. Even if one will say, okay, great. Alright, so transport in plants it is then. Alright, great. Are you guys done? Any questions, anyone? Anything which is not clear? I hope it's clear. Plant nutrition. Any questions, anyone? All right, all great. Okay, rest of you, clear? Done?
Yeah.
Okay, great. All right, then that's all for today. I'm going to see you tomorrow and we are going to start transport in plants, which is chapter number 8. All right, then see you then.